Mitosis and meiosis are the processes of cell division and reproduction in eukaryotic cells. These processes are a small part of the cell cycle. Most of the time, in the larger fraction of the cell cycle, the cell is in interphase. This phase is where most cell activity occurs. Interphase has 3 steps, the first of which being called G1, where the cell sort of lives its everyday life and grows bigger and bigger. When the cell gets too large, it doubles in size and number of cytoplasmic components, it must begin its preparation for division. The second phase of interphase is the S phase, in which DNA replication duplicates the chromosomes. In the final stage of interphase, called G2, the cell prepares to divide. As the cell cycle begins one of the division processes, interphase is concluded and the cell cycle continues. One main difference between mitosis and meiosis is the genetic makeup of the daughter cell it produces. Mitosis produces a genetically identical daughter cell. Meiosis, which occurs only in sexual reproductive gametes, produces a genetically different daughter cell. From meiosis is where we see diversity and different traits. Mitosis and meiosis follow the same basic division processes, except that meiosis goes through 2 divisions. Also, mitosis produces a diploid cell, while meiosis produces 4 haploid cells. First, let's focus on mitosis, which has 4 stages. The first stage is called prophase. In prophase, the duplicated chromosomes start to condense, and microtubules form a bipolar spindle. Also in prophase, the nuclear envelope starts to break apart. One of the 2 pairs of centrioles move to opposite sides of the cell. Next stage is metaphase, in which spindle fibers are connected at the centromere of the chromosomes, and the chromosomes are lined up midway between the spindle fiber poles. The chromosomes are in their most condensed form. The next stage is anaphase, where the sister chromatids are pulled apart from each other by spindles. The microtubules also work to push the poles apart at this stage. The final stage of mitosis is telophase, in which the 2 identical clusters reach the spindle poles and a new nuclear envelope forms. Now mitosis is over. Finally, the last step of the cell cycle, cytokinesis, may begin. Cytokinesis deals with the division of the cytoplasm and other components, everything other than the nuclear DNA material. When cytokinesis is complete, the cell goes into interphase and the cell cycle starts again. Now there are 2 diploid daughter cells in interphase. Because meiosis occurs in the reproductive cells, which have half the amount of chromosomes, the product daughter cells of meiosis are considered haploid. With reproductive cells, you inherit pairs of chromosomes. The pairs are called homologous chromosomes because they have the same length, shape, and genes. But they code for different alleles. The first step is prophase 1, where the chromosomes condense and get together with their homologs. At this point, the homologous pairs go through crossing over, where the chromosomes exchange genes and thus become different from the parent. Also in prophase 1, the centrioles move to the opposite ends of the cell. The nuclear envelope breaks up, and microtubules grab onto 1 chromosome of each homologous pair. The next stage is metaphase 1. In metaphase 1, microtubules align pairs along the equator. Next is anaphase 1. In anaphase 1, microtubules pull the homologous pairs apart, taking them to opposite poles. Other microtubules push the poles apart. In telophase 1, 2 nuclei form. All the chromosomes are still duplicated, which means they still have 2 sister chromatids. The cell needs to break them up, so the second division will begin. Let's focus on just one division. In prophase 2, the spindle fibers move to opposite sides and hook to the centromeres. In metaphase 2, the sister chromatids are aligned in the center of the cell. In anaphase 2, the sister chromatids are separated and pulled to opposite ends. The microtubules push the poles apart. In telophase 2, nuclei forms around the chromosomes at the poles. This means when you start with 1 diploid cell at the beginning of meiosis, you get 4 haploid cells out. Again, after meiosis, cytokinesis occurs to finish the new cells. Cytokinesis differs in plant and animal cells, though. In animal cells, a contractile ring is used to pinch the cells apart and divide the cytoplasm. Plant cells develop cell plates, which become a partition to separate cytoplasm, eventually maturing into a new cell wall. Well, it's time to split. Thanks for watching.